In this video, we are going to be talking about the positive and negative regulation of transcription of the arabinose operon. Arabinose is a complex process that requires multiple proteins needing to bind to multiple binding sites. As you can see from the DNA strand, there are multiple colors. Each of these colors represents a binding site for proteins involved in transcription. In the DNA strand, the red and orange color represents the ROC binding site, blue is the O2 binding site, yellow is the O1 binding site, orange is the cap binding site, red, yellow, and orange is the I1 binding site, the white is the I2 binding site, red is RB binding site. The red, purple, yellow, orange color is RA binding site, and the purple color is RD binding site. So now that we talked about the binding sites in the DNA, we're going to introduce you to the proteins that bind to these specific sites. The first one is going to be the arabinose sugar. This sugar will bind to the activator called the RSC protein. Another important activator is the cap protein. These activators are then able to recruit the RNA polymerase for transcription. Now that we talked about the key factors in arabinose transcription, we are going to show you what happens when arabinose is not present in the environment. What happens is the RA protein binds to the RO site. Another RA protein comes in and binds to the RA site facing inwards to the other RA protein. The binding pockets on the RA protein get attracted to each other, forming a curve on the DNA strand. When the binding pockets have fully bound together, they prevent the cap protein from binding to its sites. This prevents the recruitment of RNA polymerase, preventing transcription. However, if arabinose is present and glucose is absent, the sugar will bind to the RA protein, causing a conformational change and allowing it to bind to the I1 site and the I2 site. This allows the cap protein to bind to the cap site, which then signals the RNA polymerase to come in and transcribe the arabad operon for arabinose catabolization.